Hello everyone, my name is Caleb Lambert, and today I'll be helping you explore a molecular technique in cellular immunology called MHC Class 1 immunopeptidomics. We will focus on the methodology from Figure 1 of the 2023 study by Owen Letty and his collaborators. This technique is essential because it allows researchers to identify exactly which fragment of a pathogen, in this case MTB, the immune system actually sees when they are displayed on the surface of an infected cell. Without this, scientists would just be guessing at what makes a good vaccine. To understand how they identify these fragments, we can first look at the Protocol 2 workflow in Figure 1a. They describe this as an optimized BSL-3 compatible protocol designed to handle the safety requirements of working with live MTB while also maximizing the sensitivity of peptide detection. Step 1 is infection. They infect primary human macrophages for 72 hours, and this quote-unquote Goldilocks window is long enough for the cell to process the bacteria, but also short enough to avoid excessive host cell death. Step 2 is lysis. They broke open the cells to create a lysate, which is like a molecular soup of everything inside the cell. And step 3 is immunoprecipitation, or IP. Here they used a pan-MHC1 antibody called clone W632. This antibody was conjugated to physical beads. Imagine this lysate is a messy mixture. The filter acts like a capture mechanism. The W632 antibodies on the beads grab the MHC peptide complexes. As you pour the lysate through, the filter captures the beads, the MHC complexes, while the cellular debris passes through. Step 4, elution and C18 extraction. Before you analyze the beads, you should elute or wash the peptides off the MHC molecules using acid. Then use the C18 column or solid phase extraction to separate these tiny peptides from the larger MHC proteins and salts. Step 5 is fractionization. They use reverse phase LC to group peptides by hydrophobicity, reducing the complexity of the sample before the last step, step 6. And step 6, identification via LC-MS/MS. Finally, they use tandem mass spectrometry to determine the exact amino acid sequences by measuring their mass to charge ratios. Looking at figures 1b and 1c, we can see that the results of this search were like looking for a needle in a haystack. In figure 1b, the MTB peptides make up less than 0.1% of total unique peptides that were identified. Most of what the macrophage presents is either self or human. Because these bacterial hits are so rare, they performed manual inspection of the mass spectra and SIR-quant validation using synthetic standards to ensure that no false positives were reported. In Figure B, look at donor B. They treated these cells with I and F gamma. And while the absolute number of MTB IDs went up slightly, the percentage relative to the host peptides remained extremely low. Now look at Figure 1c, because it shows the validated hits. For example, the protein ESXA produced the key sequence down below. Go back to the figure, and in the green detected boxes, you can see that this specific peptide was presented by donors B and C. This confirms the macrophage successfully processed the bacterial protein. This method allowed researchers to prove a major biological point in Figure 1D. Substrates of the type 7 secretion system are massively overrepresented. While T7SS proteins are a tiny fraction of the MTB genome, they dominate the immunopeptidome. Essentially, if MTB secretes it via T7SS, the immune system is much more likely to see it. Before we wrap up, consider this. Why is the W632 antibody the quote-unquote VIP of this experiment? I'll give you a few seconds to answer. All right. Without its specificity, the LC-MS/MS would be overwhelmed by the 99.9% of human peptides, and we never find the bacterial sequences. Lastly, I wanted to reiterate this take-home point. MHC1 immunopeptidomics is a high-resolution tool that moves us beyond prediction and into direct observation of antigen presentation. Thank you so much for listening to me as I told you about this technique.